Hey guys, this is your learning assistant for Biology 1009. Today I'm here to talk about prokaryotes and eukaryotes. So yeah, prokaryotes versus eukaryotes. The left picture looks like a bacterial cell, which is the most common example of prokaryotes, and then the right picture looks like an animal cell because it has a round shape. So think about eukaryotes as like the most classic image of a cell. So when you ask a little kid what does a cell look like, I'm pretty sure they will be they'll be thinking imaging of this image. And think about prokaryotes as a cell without any organized nuclei or organelles. So those are two big difference because this one has no nucleus, but this one does have a nucleus. So let's talk about these two things in cell size. So prokaryotes are 0.2 to 2 micrometers in diameter, and eukaryotes are 10 to 100 times 10 to 100 micrometers in diameter. So relatively speaking, eukaryotes are way bigger than prokaryotes. And cell wall. So prokaryotes do have cell wall. They have two types. First one is a gram-positive cell wall, which has a thick layer of a peptidoglycan, and then the second one is a gram-negative cell wall, which has a very thin layer of peptidoglycan. So what peptidoglycan is that it is a basic component of cell wall that is composed of sugar-based polymers and amino acid. And eukaryotes do have cell wall, but only the plants and fungi have cell wall. No animal cells have one. And here's a picture of gram-positive and negative. Positive has a thick layer of a peptidoglycan, which is in red color, and here gram-negative has very thin layer of a peptidoglycan. And things about prokaryotes. So prokaryotes do not have an organized nucleus, so the DNA is in it can be found in a plasmid and or nucleoid. So nucleoid. Let's talk about nucleoid first. Nucleoid is an irregularly shaped region within the cell that contains most of the genetic material of prokaryotes. But then plasmid also has some genetic material. This is um, this is also found within the cell, but it is physically separated from a chromosomal DNA. So people call it as a not a true DNA, but technically because it has a genetic material in it, I would call it a DNA. So anyway, so plasmid and nucleoid, they both have a DNA for prokaryotes because this one doesn't have an organized nucleus. Oh, and by the way, plasmid does replicate by itself. So that's an interesting thing as well. Prokaryotes do have ribosomes, but smaller than the ribosomes of eukaryotes. The prokaryotes do have a hair-like structure, pilus. It does very interesting job. Pilus is a bridge or a connection between a bacteria and another bacteria. It helps exchanging the genetic material between them. So it's a bridge connection to exchange some DNA stuff. Prokaryotes do have cytoplasm and plasma membrane, but prokaryotes do have cytoplasm, but it does not include any cytoskeleton, which is a structure to maintain the shape of the cell. And it does divide. Prokaryotes do divide through binary fission. It doesn't do any mitosis or meiosis, so it only divides itself by binary fission. And things about eukaryotes. Eukaryotes contain its genetic material, DNA, in a nucleus. It has a cytoplasm consisting of cytoskeleton, component of protein synthesis. So after this video, there will be a short, concise video talking more in detail of eukaryotes by comparing and contrasting animal cells and plant cells. But for now, I'll be just overall generally talking how the eukaryotes make protein. So the first step will be nucleus. So at nucleus, DNA is transcribed to RNA, and then RNA is translated to protein. And the second one is a rough endoplasmic reticulum, rough ER. So it has ribosomes on its surface, and translation of the protein happens on the ribosome. And then a third transport vesicle comes off. So this is like a bus or taxi. After the protein is made, it transports to the Golgi apparatus. So the protein, after protein is made, it is transported to a Golgi apparatus in transport vesicle. And Golgi apparatus, once a protein reaches the Golgi apparatus, it moves through this organelle and is modified with sugars or glycoprotein. And at last, transport vesicle comes again. So the glycoproteins, the sugars that were modified, depart from the transphase, not the cis, transphase of the Golgi apparatus, and are transported through a transport vesicle to their destination within the cell or to the plasma membrane, where they are secreted from the cell or fuse with the membrane. So so nucleus, rough, ER, transport vesicle, and Golgi apparatus. These four things are the big, I would say, steps. So these four things are big four steps of protein synthesis of eukaryotes. So and yeah, this is pretty much what I have for today. And yeah, thank you.